Angiosperm reproduction is an uncompromising, highly engineered manufacturing process hidden within the flower. Its life cycle is designed to navigate the alternation of generations, the cycle where the visible diploid sporophyte houses and protects the fragile haploid gametophyte generation. The complete male reproductive whorl of the flower is known as the androecium. Each individual unit of the androecium is the stamen, composed of a long supporting filament and a terminal structure called the anther. The anther functions as an active biological factory, responsible for both synthesizing and deploying pollen. To understand how this factory operates, we must look past the external morphology and examine its internal geometry. The architecture of the anther must balance two contradictory functions. It must act as an impenetrable vault to protect developing spores, while preparing to rupture to release them. A transverse cross-section, a horizontal slice through the mature anther, reveals its symmetric internal organization. The anther is bilobed, consisting of two distinct left and right halves. Each lobe contains two subcompartments called theca, separated by a longitudinal groove. This four-chambered layout makes the anther dithecous. At the extreme outer corners of this cross-section are four distinct circular chambers. These are the microsporangia, the specialized sacs where microspores develop. An anther possessing these four chambers is anatomically classified as tetrasporangiate. This bilobed, dithecous, tetrasporangiate blueprint provides the universal spatial framework required to study the microscopic wall layers. Focusing on a single microsporangium, we map its boundaries outside in. The outer three layers, epidermis, endothecium, and middle layers, provide mechanical protection. The outermost boundary is the epidermis, a protective cellular shield. The second inward layer is the endothecium, where cells develop fibrous cellulose thickenings. These endothecium cells are hygroscopic, meaning they expand and contract based on ambient moisture levels. This contraction generates the force that eventually ruptures the anther. The third boundary consists of the middle layers, which store starch across 1 to 3 cell layers. These middle layers are ephemeral. They break down as the anther matures to feed the inner tissues. These outer layers form a mechanical housing engineered to physically rupture precisely when the biological payload inside is ready for release. The fourth and innermost wall layer, the tapetum, is the physiological linchpin of microsporogenesis. The tapetum serves as a highly active secretory nutritive tissue, providing specialized biological support rather than mechanical protection. Tapetal cells are often multinucleated or polyploid, meaning they contain multiple sets of chromosomes. This unique state results from endomitosis, a specialized process where the nucleus divides repeatedly, doubling genetic material, without the cell itself ever splitting. This dense genetic material allows the tapetum to synthesize the enzymes, hormones, and nutrients required by the developing spores. One of its most critical products is sporopollenin. Sporopollenin is a highly resistant organic compound capable of surviving extreme heat, strong acids, and severe enzymatic degradation. The tapetum secretes sporopollenin to form the exine, which is the hard outer shell of the pollen grain. As the pollen grains mature, they consume the tapetum entirely, eventually leaving behind an empty sac. The tapetum acts as the biochemical manufacturing engine that transforms the microsporangium from a sterile vault into a fertile chamber. The center of the microsporangium houses a dense mass of cells known as sporogenesis tissue. The individual cells within this tissue are microspore mother cells, also called pollen mother cells. These mother cells are diploid, or 2N, carrying a full set of the plant's chromosomes. Microsporogenesis is the biological process where these diploid mother cells undergo meiosis. A single diploid mother cell divides twice through meiosis to produce 4 haploid cells, known as a microspore tetrad. The geometric arrangement of these 4 cells within the tetrad is dictated by the plant's evolutionary lineage. Monocots typically form flat, tetragonal tetrads, while eudicots form pyramid-like tetrahedral tetrads. This programmed geometric division marks the transition from diploid somatic tissue to the haploid precursors of the next generation. Once the tetrads are formed, the anther begins to dehydrate. The tapetum and middle layers fully degenerate, having exhausted their nutrients to support maturation. An enzyme dissolves the callose walls holding the tetrads together, allowing the 4 microspores to dissociate into individual cells. These individual microspores finalize their sporopollen and exine layer, maturing into independent pollen grains, the male gametophytes. With the payload mature, the focus shifts back to the hygroscopic endothecium. As the anther loses water, the fibrous thickenings in the endothecium contract, creating high mechanical tension across the anther wall. This tension causes the final event, dehiscence. The anther wall rips open along its longitudinal slit. The microsporangium operates as a multi-layered protective nursery, a biochemical factory, and a mechanical deployment system, ensuring the survival and spread of the plant's genetic code.